radiating coloration a lot, but they also converge the radiation in their color pattern. So smelling and tasting is very important in the lifetimes of almost all organisms in the world. So it's involved in mate choice, in recognizing your own species, your own kin, and also in recognizing what is edible or not. So this is why I became interested in studying these ionotropic receptors in these butterflies. So you have different color morphs of the same species. So this is the same species, just in different locations. They look totally different. But at the same time, this butterfly co-occurs with its mimic in the same country. And in every country they look different, but they look the same, these two species. So when they court, or when the males are looking for a female, they fly around, they see something that looks like them, they approach it. But in the case when they find their own species, they approach it and they continue courtship when they come close because everything looks good. And in the case of mimicking in their mimic, it still looks good, but apparently something smells wrong because they fly away again when they come close. So from that, I deduced that there should be more than just visual stimuli for courtship, and chemical properties should be important. And that's why I started studying how ionotropic receptors evolved in this genus. So ionotropic receptors were discovered only in 2005 after people in Drosophila couldn't figure out what these sensory neurons were sensing because they didn't express any gustatory or olfactory receptors. So they found this third class of receptors. And then they found out that these are actually the oldest receptors. This is the first gamma reception system evolved in insects because it already evolved very early in Protostomia. So this was when Protostomia were still more like snails and worms. So these were a system that does both tasting and smelling. Like chemically, they're very similar how it works. And I wanted to know if there was any duplications in by butterflies that would facilitate them to recognize their own species. So what I did, I used publicly available data all the way. So there was a genome already published. There was RNA-seq data of the 3 sensory tissues, which are legs, antenna, and the proboscis. So the proboscis and legs are more like they touch things with it, but they also touch things with the antenna, so they smell and taste with all 3 sensory organs. So smelling and tasting is not like very, it's like the kind of molecules that they taste or smell make it different, but the way they do it with the same organs. So there was already data available of ionotropic receptors in these 3 other species, which I used to compare, which are 2 Lepidoptera and 1 Diptera. And those genomes are also publicly available, and so I used it to annotate the IRs in Melpomene. After that, I went to the whole genus and got like 10 out of 50 species and annotated the ionotropic receptors also in these, using freely available low quality sequence data, so I could make a very primitive genome and then filter out my genes using BLAST. So what I found out is that our butterfly that I studied, which is Melpomene, which has the genome, has 31 ionotropic receptors, which is more than Bombyx and Danaus, and a lot less than Drosophila, because Drosophila have these ionotropic receptors that are mostly in their gustatory tissues that seem to taste things mostly, and which is this green blob, which it has no representative in Lepidoptera, while you have all these genes that are not colored here that are very conserved and are mostly expressed in the antenna, and they have like conserved copies between all 4 of these, most of them, and there's like little exceptions everywhere. And then you have these 2 clusters of genes that only occur in Lepidoptera, but have no equivalent in Diptera. And then here you have 1 gene that is present in both butterflies, but not in the moth. And then here, these in red are the most interesting ones because these only occur in Heliconius and have undergone multiple duplications in Heliconius. So then here you can see that the genes that undergone duplication, the only one I could find sequence data from, because they're so very similar and I did the de novo expression analysis, I found only expression for one gene, and they're more highly expressed in the proboscis and the legs, but not in the, like, much lower expressed in the antenna, which suggests that they could have more of a tasting role, but that's definitely not definitive. And then I went to look at all the other butterflies in the genus. Now I had like clear annotations of my ionotropic receptors. I could easily find them in the other species. And basically, here you have these duplications that I found in Melpomene compared to the other Lepidoptera. And here are some genes missing that are probably either absent in the genome of these two species, or they're just not found in the genomic data that I had. So if you zoom in to these two genes, so in Bombyx and Danos, these are just two genes. And in some of my butterflies, there's 7, I also have 5, 4, and 3. So there's like, within this genus, there's been a lot of duplications going on, and especially in this group. And that's very interesting, because these are the only 3 species that still hybridize. So these guys share genes between these 3 species. So here you have, like, this is all Cythno, and here you have Timaretta. So what is the theory is that Cythno and Melpomene split apart like 5 million years ago, and then after that, Cythno hybridized with Melpomene to make a Timaretta. So and then this Timaretta had enough Cythno genes to stay separate from Melpomene, but they adapted the wing pattern of Melpomene. So here they look like Melpomene. So they're basically Cythnos that look like Melpomenes and co-occur with Melpomene. So for this to happen, like they share all the genes, they lay eggs in similar host plants, but still genomically they stay separate. If you look at the whole genome level, they're clearly different. But when you see them in the field, Melpomene and Timaretta look totally the same. So looking at this and seeing that they have like these 7 genes, these extra duplications, that could be because they needed to better distinguish their own from their mimic. Because their mimic, instead of, in the beginning I showed the main mimic, which is Errata, which is very distantly related, as a genome that's twice as big, so there's a lot of very little potential of confusion, while these guys are so closely related that they can and will produce fertile offspring together, but they still remain separate, even though there's gene flow between the species. And my theory is that these duplications that are found in these ionotropic receptors could be facilitating pre-zygotic isolation between these species. So what I found on ionotropic receptors is that some are very conserved, even between Diptera and Lepidoptera. Others, on the other hand, have undergone duplications and facilitate species-specific needs. And especially the IR68 are very interesting because they show an increased copy number within the genus Heliconius, which correlates with their adaptive radiation. And they are also the only IRs that are higher expressed in legs and proboscis versus antenna. And this genetic variation, I hypothesize, could very well be involved with mate choice in the Heliconius lineage, and that would be something I really want to study after this. And I would like to thank people at the University of Puerto Rico in my lab, and my collaborators, Chris Jiggins and Adriana Briscoe. And are there any questions?